This research study was designed to investigate the effects of curcumin on cell proliferation and apoptosis in mammalian breast cancer cells. Currently, breast cancer is the most prevalent cancer worldwide. It occurs when some breast cells begin to grow abnormally. There are many plants reported to have anti-cancer properties. Turmeric is one of those plants. Curcumin is extracted from turmeric and is an agent that prevents cell proliferation by blocking the cell cycle and accelerating apoptosis. The hypothesis of this experiment was that curcumin extracted from turmeric possesses anti-cancer properties and will inhibit cell proliferation and induce apoptosis in breast cancer cells. For my methods, I started by extracting curcumin from dry turmeric through supercritical fluid extraction. CO2 was used as the main solvent for the extraction. The extract was then rotary evaporated and analyzed with UPLC. The breast cancer cell line used for this experiment was MDA MB231 cell line, along with a non-cancerous cell line CHOK1. The cells were cultured and plated, followed by treatment with 5, 10, 20, and 30 micrograms per ml curcumin. For cell viability, the cells were treated overnight with curcumin followed by MTT assay. The apoptotic activity was investigated using ethidium bromide and acridine orange acid. The cells were treated with 5 and 10 micrograms per ml curcumin. The EBAO dye was prepared with 100 micrograms per ml ethidium bromide and 100 micrograms per ml acridine orange in 1x PBS. The cells were stained with the dye and then viewed and counted using DAPI, GFP, Texas Red Excitation Filter in Citation 5 software. For RT-qPCR, the cells were treated with 5 and 10 micrograms per ml curcumin, followed by RNA extraction and cDNA synthesis. Annealing temperature was then optimized, and then the samples were run for the experimental run. For the results, I got higher amount of 784.30 mg per liter curcumin present in the sample, which was extracted using co-solvent, and only 28.75 mg per liter curcumin in the sample, which was extracted without co-solvent. The cell viability assay showed that highest cell viability of approximately 80% was observed in both the cell lines treated with 5 micrograms per ml curcumin, and the lowest cell viability of approximately 35% was observed in both the cell lines in cells treated with 30 micrograms per ml curcumin. The EBAO assay showed that the majority of cells underwent apoptosis or necrosis. The PCR result was unexpected because the BAX gene only upregulated in cells treated with 10 micrograms per ml curcumin in MDA MB231 cell line and cells treated with 5 micrograms per ml curcumin in CHOK1. The SFE extraction shows that more curcumin can be extracted with co-solvent. The cell viability results shows that cells die when treated with curcumin irrespective of whether they are cancerous or non-cancerous. The PCR result was unexpected because we did not wanted our BCL to express, but it did. In conclusion, the hypothesis was met because the results shows that curcumin possesses anti-cancer properties and inhibits cell proliferation.